Mic check.
Mic check.
Hello, everyone. Welcome to Antibuddies Journal Club. Today is our 27th session, and well, also unfortunately, the last session of the year before we take our winter breaks. Joining me today is our a special guest with us, Dr. Gustav Christopherson. I'll tell you guys a little bit about Gustav. Gustav's research interests are mainly focused around local immune regulation in the pancreas during the context of autoimmune diabetes. His group studies this using animal models, human tissues, and human clinical imaging. Apart from research, Gustav also likes to be outdoors, hiking, skiing, or biking. Gustav, it's an honor to be here joining this discussion with you. Welcome to Antibodies.
Thank you. It's great to be here, and thank you for the invitation to present here uh, and talk to you, antibodies. Um, yeah, is it time to start?
Oh, yeah.
Should I go on with the presentation?
Yeah, we can begin the
Um,
discussion now.
yeah. So today I will present a paper we published about a year ago, so it's not um, super fresh, but I hope it's still interesting. It's so as Jatin said, I'm interested in the local immune regulation in the pancreas, and at the time when this project started, I was a postdoc in Matthias von Herat's lab in La Jolla at the La Jolla Institute for Immunology, and we were working a lot on the antigen specificity of immune cells and sort of what antigen specificity and sort of non-specificity, what the role of that had to do with T cell trafficking and sort of the impact on the autoimmune response. But then we got interested also in sympathetic nervous signaling, and I will touch a bit about how we got into this later in the talk. But we so did a few, I guess, rather simple experiments where we sort of, to our surprise, saw a fairly large impact in sort of a large impact on the autoimmune response, like from sympathetic signaling in the pancreas. So yeah, as Jatin said, now I'm an associate senior lecturer at Uppsala University in Sweden, and I'm also affiliated to the Science for Life Laboratory, or SciLife Lab here. So this study was performed both at the La Jolla Institute and in my lab at Uppsala University. So just some introduction to sort of type 1 diabetes and you know, what we're looking at here, and of course at the center of attention for all of us working on type 1 diabetes is of course the pancreas, situated right in the center of our bodies, and more specifically the islets of Langerhans, that are these miniature endocrine organs that are scattered throughout the pancreas. They are very small, about 100 to 200 micrometers in diameter, and they make up about 1 or 2% of the total sort of, pancreatic mass. So they're not a large fraction of the pancreas, and they're very sort of scattered in the pancreas. So if we look closer at the pancreatic islet, it consists, of course, of different endocrine cells, alpha, beta, delta cells, etc. And when in type 1 diabetes, there is some kind of trigger, some triggering event that we sort of don't know what it is at the moment. But something that allows for the influx of autoreactive T cells into the islet, and for these autoreactive T cells to start killing the beta cells. And after a while, this sort of immune environment gets more and more complicated. We have more and more T cells invading other kinds of immune cells, macrophages, dendritic cells, B cells, neutrophils, etc. So this will, after a while, turn into a fairly sort of complex environment. And after a while, most of your beta cells will be killed off, and the person with type 1 diabetes will no longer be able to sort of regulate their blood glucose. But one issue with studying type 1 diabetes is that the patient shows up at the clinic first when the islets look something like this, when you have lost a fairly large amount of your beta cells. And so most of the interesting events, the triggering events and these sort of initial immune events have already happened when the patient comes to the clinic and sees the physician. So one problem with studying type 1 diabetes is that sampling a patient when the islet looks something like this, and when most of the immune response has already taken place, that likely doesn't tell us much about the early events in the disease. And another problem with the pancreas as an organ is also that you cannot biopsy it. We can't take biopsies of the pancreas of a person with type 1 diabetes. So we're sort of limited to mostly sort of blood samples and to some extent sort of clinical imaging modalities such as PET and CT and MRI, etc. So it's a fairly sort of difficult disease to study in the human. Just to sort of point out a couple of cells that we in my lab are interested in that are present in the islet is the macrophage, the resident macrophage in the islet. That is a bit special, and I will touch upon that later. And also the nerves that come into the islets of Langerhans. And of course, also a possible interplay between the nerves and the immune cells, and in particular in the macrophages here. So these difficulties in studying some human type 1 diabetes, we try to sort of circumvent them a bit in my lab by using mouse models. And even though they're not sort of perfect for studying the disease, they can tell us quite a bit about the basic immune regulation as well. So one way that we look at this is by sort of actually sort of looking at what happens in autoimmune diabetes in these mice. So what you see here is an islet of Langerhans, or at least what used to be an islet of Langerhans. These yellow cells are the insulin-producing beta cells. And this islet used to be sort of filled with these yellow cells, but now you can see that most of them are gone. They have been killed off by these pink CD8 positive T cells that you can see running around in and around the islet. they will likely sort of kill off these last few remaining beta cells within the next few hours here. So we do intravital imaging of the pancreas and the islets of Langerhans to see interactions between immune cells and immune cells with their target cells, i.e. the beta cells, and also look at sort of trafficking of immune cells, etc. And this can tell us quite a lot about sort of what goes on in the pancreas and how different treatments affect the immune response locally in the pancreas. And we do this by using a 3D printed vacuum window that we have sort of modified ourselves. You can just see a drawing. We published the drawings of this a couple of years ago. And this is a version with a heated ring so that we can sort of keep physiological temperature also during long imaging sessions. And we're, of course, not sort of limited to just looking at the pancreas. Here are just some examples of these are different kinds of Tregs in the spleen, for example, just labeled with different fluorophores. This is, again, Tregs, but in a lymph node. So we can look at many different organs, lymphoid organs, and in other projects, look at intestine, muscle, etc. So it's, we find this to be a pretty powerful tool to study sort of immune events in different parts of the mouse body. And then moving back to sort of human type 1 diabetes, and just to set things in a bit of a more sort of context. So this graph just has beta cell mass on this axis and sort of age or time on this axis. And what we can see in human type 1 diabetes is that there is a period before sort of onset of disease where the persons are autoantibody positive. So they have antibodies specific for antigens expressed in the pancreatic islet. So what then must have occurred is that there is some kind of triggering event that we don't know about yet. If it's some kind of infection, viral infections have been discussed a lot in type 1 diabetes, or stress, or some other sort of environmental, dietary things that could have triggered an autoimmune reaction. We have autoantibody positivity. We can see also hyperexpression of MHC class 1 on beta cells. So there is a period before sort of clinical onset of disease where things are starting to happen. And then the immune response is sort of seemingly increased, and we come to a point when the amount of beta cells, or the beta cell mass in the pancreas, can no longer support the regulation of the blood glucose, and so the patient shows up at the clinic and is diagnosed with type 1 diabetes. And we don't know exactly sort of how this occurs, how this sort of happens over time. This is one model that is in a kind of sort of relapsing remitting pattern, that you have sort of waves of immune attacks directed towards the beta cells over time. We don't know if that's the case, but this is sort of just one model, one hypothesis of what happens. So we're then interested in whether nerves and nerve signals are active in these sort of early stages of disease. And a striking feature of human type 1 diabetes is that you can see a certain lobularity of sort of islet inflammation in the pancreas. So this section of a human pancreas from a type 1 diabetes patient has been stained for MHC class 1 here in red, where we can see that some islets hyperexpress MHC class 1 and others don't. And one can also see sometimes that certain lobes of the pancreas are seemingly unaffected by the autoimmune response. You have remaining beta cells, a fairly low level of immune infiltration, whereas a neighboring lobe can be completely devoid of beta cells or have a lot of immune infiltrate in their beta cells or in their islets. So this is something that's very intriguing, and we don't know why this is. Why is not the whole pancreas affected by the autoreactive response at the same time? And this has then been hypothesized to be dependent on perhaps blood vessels, sort of how the vasculature branches into the pancreas, or how the nervous system branches into the pancreas. Yeah, I see we have a question, Jatin.
Yeah, Gustav, I was wondering, is this, when you said in human islet inflammation, is it not seen in mice at all?
Well, I have one example here. So it's not as prominent at all in mice, but we can see, sometimes for a very short period. So here you see is also in an Intravital movie. We have CD8 positive T cells in one lobe of the pancreas, but you can see that the neighboring lobe is doesn't have many infiltrating CD8 T cells at all.
Okay, so it is similar in mice as well. Then.
It might be. The problem with the mouse models is that the autoimmune response is very sort of aggressive and rapid. So I think even if the same thing would happen, it happens very rapidly. So it's very difficult to see this lobularity. But this sort of just an example of that, there might be something similar going on also in mice. Yeah, so we have this lobularity, this sort of difference from sort of lobe to lobe in the pancreas, and we don't know why that is, but innervation has been hypothesized to be sort of one reason for this. And looking at other autoimmune diseases, we can also see patchiness in other autoimmune diseases, such as vitiligo. You have a patchy loss of pigmentation in the skin. This is also interesting. So why is not all pigmentation lost at the same time? Here's also a very interesting sort of symmetry in the majority of vitiligo cases. As you can see here, there's the very symmetrical sort of across the two sides of the body. And why is that symmetry there? Here also a sort of a nervous system component has been proposed, since the nervous system also is sort of symmetrically distributed across the body. Same thing goes for rheumatoid arthritis. Most often, sort of both joints on both sides of the body are affected at the same time. So there's also symmetry there, and why would then sort of both your knees be affected at the same time, and not sort of first one of them and then perhaps another joint in the arm, etc. But seems very symmetrical there as well. So we have these sort of observations also in other autoimmune diseases. So what triggered this this study that we're talking about today is, I told you we were working on a completely different project, but then this issue of the Scientific American landed in the lab, and it was about these sort of bioelectronic medical devices. And we looked into this in type 1 diabetes, and we saw that there wasn't too much done here. And we started thinking that we could, you know, with a few simple experiments, see if there's sort of any effect in the model that we're using. So this is what triggered it. And looking at some neuroimmunology, or the interaction between the nervous system and the immune system, this field of study has boomed during the last few years, and there's been a lot of papers coming out showing an effect on the interaction between the sympathetic nervous system and immune cells in homeostatic processes such as intestinal peristaltics and intestinal barrier function, where signals from the nervous system to intestinal macrophages can affect both barrier function and peristaltics. We have in adipose tissue effects on temperature regulation and lipolysis. If we look at pathophysiology, there are neuroimmune effects in multiple sclerosis, in rheumatoid arthritis, and there are also some papers on autoimmune diabetes. So we decided to look more closely into this in the models that we were using to study autoimmune diabetes. And we can see that islets in mice are highly innervated by neurons from the sympathetic, parasympathetic, and sensory nervous systems. So we have a lot of nerves here. We find these also in human islets. However, the sort of innervation pattern looks a bit different, but the nerves are there. We can see also these are some just unpublished images from some mass spectrometry imaging we've done. So IAPP marks islets in this pancreatic section. You can see here in a consecutive section that where we have the islets, we also have our the major concentration of norepinephrine sort of at the sites where we have islets. So the islets are, there is an active sort of sympathetic nervous system in the islets in these mice. And going back to the human situation, there was a paper in 2016 showing that there's a marked loss of nerves in early type 1 diabetes. So they looked at short duration type 1 diabetes, where they could see very low levels of tyrosine hydroxylase positive nerves in the islets. And this then marks sympathetic nerves. So this was very obvious in this study. And now fairly recently this year, there was another study that came out that didn't see this very marked decrease. However, what was interesting here was that they saw a decrease in the amount of sympathetic nerves already in these autoantibody positive individuals that have not yet developed type 1 diabetes, but they have a high likelihood of developing it later in life. So there might be a sort of somewhat of a disturbance in the innervation of islets in the pancreas already in early type 1 diabetes, and also perhaps then even before onset of disease. So the model that we used to study this was the LCMV-RIP-GP model of autoimmune diabetes. So how this model works is that it's a transgenic model where the beta cells of the islets have been made to express a viral glycoprotein on their surface. And it's the glycoprotein from the LCMV virus. And what we do then is to infect these animals with the LCMV virus. The mouse will mount an immune response against the virus. And of course, this glycoprotein expressed by the virus, but then will, of course, also mount an immune response against the glycoprotein on the beta cells. So in about 2 weeks' time, most of the beta cells in these mice will then be lost in a sort of induced autoimmune-like fashion. So this was the model we used, and we then just wanted to see whether the sympathetic nervous system had an effect in this model. So we did this in a few different ways. We denervated the pancreas surgically. We cut one of the sort of sympathetic nervous bundles that innervates the pancreas. We used a sympathetic neurotoxin, 6-hydroxydopamine, that depletes all sort of peripheral sympathetic nerves. And we also used antagonists for alpha and beta adrenergic receptors. So what this looked like when we did the denervation experiment. So we have here surgical denervation in green, 6-hydroxydopamine, this neurotoxin in red, and then just sham surgery in blue. So what you can see with the sham surgery, these mice, all of them turned diabetic within 2 weeks. But then to our surprise, these denervations were fairly effective in protecting the mice from this autoimmune diabetes. And here we should remember that this is a fairly aggressive model of autoimmune diabetes. So this is a monospecific model of autoimmune diabetes that is generally fairly difficult to halt the immune response in. So we were pretty surprised by this, that sort of just interfering with innervation like this could halt this autoimmune response. And then we looked at also the what receptors could be involved in this, and we used Pitocin, that is a alpha-1 adrenergic receptor antagonist, propranolol, a non-selective beta blocker. And as you can see, our control sort of vehicle treated animals turned diabetic again within 2 weeks. The beta blocker propranolol had no effect on diabetes, whereas the alpha-1 blocker Pitocin also was protective to sort of a similar extent to the what we had seen in the Denervation experiments. So we were pretty surprised by this, that the effect was this large. So we had a look to see whether these treatments were affecting the response to antigen in these CD8 positive T cells. These are the sort of main effectors in this model. But we can see that the Pitocin and 6-hydroxydopamine treated mice, these CD8 T cells, they were producing interferon gamma to a similar extent as the control mice. So this wasn't due to any sort of suppression of the antigen-specific response. We looked at peripheral immune cells, CD4 T cells, CD8 T cells, CD11 B cells. And even though at sort of certain time points during onset of disease, we have some differences between the groups, we couldn't find any sort of obvious pattern that could explain what was going on in the pancreas. So this was a bit puzzling to us, that we couldn't see sort of any sort of obvious sort of protective effects either on CD8 positive T cells or in sort of circulating immune cells in these mice. So then we turned to intravital imaging and had a look at what's actually going on in the pancreas. So this is one of the control mice. You see here 2 islets. You can see they are infiltrated by many macrophages in green. We have antigen, sort of beta cell specific CD8 positive T cells in red. So these are transgenic T cells as well that have a transgenic T cell receptor that recognizes this glycoprotein on the beta cells. You can see in the control situation, many macrophages, many T cells in the islets. And these mice will then eventually turn diabetic. But then when we look in the denervated animals, so here you have one of the islets here, we saw that they were not infiltrated by macrophages to the same extent. This is more or less what it looks like when you look in a healthy mouse. When you look at the macrophages, these are mostly the sort of resident macrophages that are always there. But we did see quite a few CD8 positive T cells around the islet. But what we saw was that the behavior of these CD8 positive T cells was a bit different. So when we compare to the control situation, we see that these CD8 positive T cells here are running around a lot more. They migrate with higher speeds and higher distances, which then might point to that they are sort of looking for antigen, or looking for sort of target cells or antigen presenting cells to interact with, but somehow they don't find this, since most of these denervated animals actually remained non-diabetic. So looking at this and quantifying data from the imaging, we see that the numbers of macrophages were lower in these alpha-1 blocked mice and the 6-hydroxydopamine treated mice. The numbers of CD8 positive T cells were significantly lower in the 6-hydroxydopamine treated mice. And then when we look at behavior of the CD8 positive T cells, we see here then that they migrate a lot faster in these denervated mice, and migrate much longer distances, which then might point to that they're just not sort of interacting with their target tissue. And then we try to come to some kind of mechanism behind this. And when we look in the islet of Langerhans, we see that a lot of the nerves, or sympathetic nerves at least, are in very close contact to macrophages in the islets. So almost all resident macrophages in islets are in contact with a sympathetic neuron. And macrophages in islets are interesting since they are in an inflammatory state. So if you look here, I just pulled a figure from another paper that macrophages in islets are mostly of an sort of inflammatory sort of M1 type of macrophage. And you can see here that macrophages in islets do not express MMR, for example, whereas macrophages in the exocrine tissue surrounding these islets, they do express MMR to a large extent. So macrophages in islets are a bit different. So we were thinking that, okay, what if these resident macrophages are some kind of sort of neuroimmune relay here? And we also did just a simple experiment in just depleting macrophages from these mice before inducing diabetes in them. So what we can see is that mice receiving clotted liposomes to deplete macrophages, they do not develop type 1 diabetes. And this has been seen in other mouse models of type 1 diabetes as well, such as the NOD model, that when you deplete macrophages, the mice do not turn diabetic. So the macrophage is important for inducing this immune response. So we wanted to look a bit closer into the islet resident macrophages and how they possibly could sort of affect the immune response. So we looked at their expression of adrenergic receptors. So we isolate macrophages from the exocrine tissue and from islets, and we perform flow cytometry on these. And we can see that macrophages in islets seem to express more of adrenoceptor alpha 1, a bit more of adrenoceptor beta 2. So they do express the receptors for epinephrine, norepinephrine. And they seem to do so to a bit larger extent than macrophages in the exocrine part of the pancreas. And when we take out these macrophages from the islets, we put them in culture dishes and stimulate them in vitro with norepinephrine. We can see that when we stimulate with different concentrations of norepinephrine, one concentration that's supposed to be a bit more sort of immune anti-inflammatory concentration, and another concentration that's sort of more inflammatory. And this depends a bit on sort of which concentrations these two receptors respond to. And we can see that yes, these islet macrophages do respond to norepinephrine, and that the response to these different concentrations does differ. And we can see also when we block this in vitro with using predsosin, this alpha-1 blocker, or butoxamine, a beta-2 blocker, we can sort of recapitulate somewhat the immune response that we think that we see also in vivo in these diabetic mice. So we, even though we didn't come to a full out sort of strong mechanism in this paper, we at least started touching upon a possible effect of islet macrophages acting as some kind of neuroimmune relay in the islet during onset of type 1 diabetes. So just to summarize, we have here shown that denervation and alpha-1 adrenoceptor inhibition halts onset of type 1 diabetes in a mouse model. This seems to be a local effect in the pancreas, since we can't see any obvious differences peripherally on immune cells. And we also then looked at these islet resident macrophages, and we can see that they express adrenergic receptors and that they do respond to catecholamines. And of course, there are still many outstanding questions here. So where does the sympathetic input have its effect? Is it locally in the islets? Is it the macrophages? Is it T cells in the pancreas, or is it the innervation of lymph nodes that has this effect? Is there a unique neuroimmune interface between nerves and macrophages in the islets? We're looking at this now in more detail. And also, of course, the human situation is islet innervation and or activity altered at pre-diabetic or early onset stages of the disease. This is also something we're looking more closely into using some clinical imaging methods. So still a work in progress, but we find this very interesting, and as I said, many other papers that have come out in the last years have shown a large effects of the nervous system on immune cells, and we see here that the same might be true also for type 1 diabetes. Yeah, just to acknowledge the people involved in the study. As I said, the study was initiated at the La Jolla Institute for Immunology with Professor Matthias von Herat and a very talented technician, Sabarnika Ratliff. And then, as I said, now I'm at Uppsala University in Sweden. These are the lab members now. Your antibody member VJ is working a lot on these projects at the moment. And so here are my current funding agencies. And I thank you for listening to this. And I hope to be able to answer any questions that you might have.
Thanks a lot. That was I have some comments, but before that, are there any other questions?
I have one question from the audience. Like, now to ask whether this phenomenon, like the exact phenomenon that the macrophages interact with nerves are present. This phenomenon is present in the other autoimmune diseases. Like, is it found anywhere else? Like, this interaction.
Yes, it's studied a bit differently in different models, but there is definitely evidence of this kind of interaction also from other organ systems. As I had there on one of the early slides, a lot of studies have been done on macrophages in the intestine, and there you seem to have a very sort of tight interaction between sympathetic nerves and macrophages, and that innervation does or sort of signals from the sympathetic nervous system does affect the activity of these macrophages.
When I first came across this study, my conception was that, yeah, I am expecting the nerves to be immunomodulatory. That was my expectation. And then this study completely changes that. So I wonder, why would we have such a system where nerves are stimulating the immune system?
Yeah, I mean, that's something we thought about a lot also, why how is this occurring, or sort of how can the nervous system know sort of exactly where to modulate the activity of immune cells. But it might be that the nervous system can put some kind of tonic inhibition that's would be sort of the normal state. That's sort of a low and constant sort of catecholaminergic signaling would sort of limit inflammation, have sort of an anti-inflammatory effect. And then sort of depending on the situation, and sort of in this case losing part of that signaling or increasing signaling, then can sort of switch the sort of inflammatory state of a particular tissue. Why this occurs, how this occurs, we don't know. I think there is at least in the case of the pancreatic islets and type 1 diabetes, that it's a very intricate process, and that we also have more than two cells involved here, that not just the nerve and the macrophage, but there's sort of a longer chain of events that leads up to this.
Okay. I was just thinking, I don't know if it's already done or this could be something, I guess, meta-analysis, looking at onset of type 1 diabetes on people who are under antidepressants or other drugs that alter your nervous system.
Yeah, I mean, that's very interesting. One observation that is out there is that there is in some epidemiological studies a correlation between type 1 diabetes onset and what they call sort of major life events. If you have some kind of crisis in your life, a major loss of a close person, or depression, etc., that seem to sort of correlate to onset of type 1 diabetes. So these stress seem to have an effect on the disease. But then your idea there with looking at sort of antidepressants and other sort of pharmaceuticals that can affect the nervous system. We have, it's not published yet, but we have looked at, for example, beta blockers, since we see this effect of at least non-protection in sort of in beta and alpha blockers, the non-protection and protection in the mouse model. We couldn't see, at least in the data that we looked at, we couldn't see much of that in humans. But I think there's a lot of sort of data available already in registries that we can have a closer look at.
Okay. This is the last thing for me. I am a little bummed out by this study because now I can no longer ignore neuronal diseases and all those papers. I used to shrug those papers off. That not in my alley. But now I'll have to read those. Thank you.
Yeah, I mean, it's we used to say that immunology is complicated, and now it gets even more complicated, right?
Yeah.
But yeah, and then there's of course the difference between what happens in the central nervous system, and there you have also sort of very intricate sort of neuroimmune events going on. But I think there's also a bit of a difference from what goes on in the central nervous system and what goes on in peripheral tissues. You have a different set of immune cells around, and different kind of innervation, of course. So there's also that sort of difference between sort of the central nervous system and the peripheral nervous system. That's also interesting. And as you say, a lot has been done on the central nervous system, but not so much on the periphery. But as I said, that more and more is coming out.
Yeah. What you said about the major life events, now it's just it makes it so complicated because not just a patient's medical history or the genetics, things that are happening in their life can affect their it's layer after layer of what can affect you. It's crazy.
Yeah, it's I mean, it's not just what your what pathogens you're exposed to. It's not just the food you eat, but also your well, more or less, perhaps your feelings, your level of stress, the sort of activity of your nervous system might have a large effect also on the immune system.
Okay. So before we wrap up the session, are there any other questions? Magda, are you clear from the audience?
Yeah, in fact, like we had this another minor question about like why there are so many nerves in and like neurons in the pancreas, but I think like this is just for the physiological reason and endocrine regulation. But if you want to elaborate on this.
Yeah, I could just I didn't touch upon the sort of the effects on the endocrine cells by the nervous system, but of course there is also an effect both from the sympathetic and parasympathetic system on alpha and beta cells and their secretion of glucagon and insulin. And that's a whole field by itself, you know, how there's a link between the central nervous system and our endocrine system. And yes, there is an effect, but also the islets of Langerhans can function sort of autonomically, and they do not need to have innervation to respond to glucose. But yeah, I mean that might be a reason for there being a fairly high level of innervation, that it's also there to sort of modulate the release and production of hormones.
Okay, I think this would be a good time to wrap up our discussion. Thanks a lot, Gustav, for this wonderful paper presentation. I'm sure everybody learned a lot. For our audience, well, as I said in the beginning of the session, this is our last journal club for this year before we go on a winter break. We'll see you guys again in January. Until then, bye bye.